Hi everyone, this is the last lecture of SCB 204, lecture 36. And fittingly, this is on heredity. So in this lecture, we're going to review some of the definitions and terms you probably learned in the lab already, such as genotype and phenotype. And you want to make sure you have a good understanding of these, because questions on both the lecture final and the lab final may, dictate what the correct answer is going to be. Okay. I've modified this lecture over the years, so some of this outline is probably not very accurate anymore. So we're going to talk more about counseling, amniocentesis, and chorionic villi sampling, and we'll give you some examples of different types of inheritance. So just some background. Most of this you know already, I think. We have 23 pairs of homologous chromosomes in each nucleus. And remember, we have 46 chromosomes, but we have 23 pairs of homologous chromosomes, 23 different chromosomes. So each pair of homologous chromosomes contains one from each parent. So you received one set from your mother, and you received one set from your father. Okay? And most of your body or somatic cells are diploid, since they have two copies from each chromosome. And these chromosomes, chromosomes 1 through 22, are called autosomes. Okay, they're non-sex chromosomes. And each pair of chromosomes have the same number of genes. Okay, the 23rd pair are the sex chromosomes, and it's either an X or an Y. And you can set up the full set of the 23 chromosome pairs in a picture and displayed in what's called a genotype. So right here on the right here, is a karyotype. Okay, and you can see all 23 pairs of chromosomes, and very tiny, but it's sort of X and Y. I think I have a larger picture of the X and Y, and each pair of homologous chromosomes have the same genes located in the same place. Okay. Now here are two important terms that you have to get correct, or else things are not going to confuse, and you're going to miss questions on both final exams. The genetic makeup of an individual is a genotype. So this tells you the actual genes that are present in that person. Okay, it doesn't necessarily tell you what they look like or what they express, but it just tells you what genes they have. The phenotype is a physical trait, and this is what we see. And likewise, what we see does not necessarily tell us what the genetic makeup is. So we need both. Okay. Now, homologous chromosomes have pairs of genes that code for the same trait. Okay, and these are pairs of genes. Okay, and any differences in forms of these genes are called alleles. Okay, so homologous genes that are not identical are called alleles. And homologous genes, as we mentioned, are located at the same location or locus of that chromosome. The location of a gene is called its locus. For example, gene for every eye color may code for blue eyes or brown eyes. Okay. So whatever eye color gene you have is a genotype. The color of your eyes are the phenotype. Now, if two alleles code for the same trait, they are called homozygous for that trait. In other words, both alleles are the same. Okay. If the two alleles are different, then they are called heterozygous for that trait. So if you have two different genes, it's heterozygous. If it's two same genes or same alleles, it's called homozygous. Now, some alleles are not equal to each other, because some alleles may mask or suppress the expression of another allele for the same gene. Those alleles that mask another gene or another allele are called a dominant. Okay. And an allele that has become masked from another gene is known as being recessive. Okay. Now, we abbreviate the dominant alleles by using capital letters, sort of like big B and big B. Okay. Recessive alleles are represented with lowercase letters, like small b, small b. So here's small b, and here's big B and big B. So the big B's are the dominant alleles, and the little b's are the recessive alleles. So let's now look at more closely at genotype and phenotype. Okay, so an individual's genetic makeup is called the genotype. It describes what alleles of genes they have. The physical expression of an individual's genotype is called the phenotype, and this is what is physically seen in terms of a protein or a feature. Now, there are different types of patterns of inheritance. Okay, some phenotypes are linked to a single gene pair, which are very simple ones, but by far most are by multiple alleles or several genes, and they're very complicated. Okay, let's begin with something simple, like with dominant and recessive traits. We have what are called autosomal dominant recessive inheritance, and this refers to the interaction of alleles strictly dominant or recessive. So when you have an allele, it has to be dominant or recessive. Of course, the dominant will mask the recessive alleles. So here are some examples of dominant and recessive traits. So we have autosomal dominant traits, and we have autosomal recessive traits. The autosomal dominant traits are things like freckles, dimples, and astigmatism, free earlobes. These are examples of autosomal dominant traits. Autosomal recessive traits are things like no freckles, lack of dimples, non-distorted vision, or without an astigmatism, and attached earlobes. These are all autosomal recessive traits. Okay. Now, we can use a tool called a Punnett Square to predict possible genotypes of offspring between parents. A Punnett Square looks sort of like a tic-tac-toe box almost. And either on the tops and the sides, we put the possible alleles that could be present, and within the boxes, we could fill in all the possible permutations of those alleles. Okay, so for example, here we're looking at different alleles for dimples. Okay, so in the very top we have the mother's genotype, and this mother's genotype is heterozygous because she has a dominant gene, big D, and recessive gene, little d. Okay, so having dimples is dominant with big D, lack of dimples is recessive with little d. So the mother is heterozygous, big D, little d. On the left side here, we have the possible alleles produced by the sperm from the father, and in this case, he is also heterozygous, meaning that he has one dominant allele, big D, and he has a little d allele here. And we fit them on either side of the Punnett Square, and we just cross each other to make sure that we make every permutation possible. So for example, we have big D and big D here, that produces big D right here. We have a big D here from the father and a little d from the mother, so we have big D, little d. We have big D from the mother and little d from the father, so this is big D, little d. Finally, we have little d from the mother and little d from the father, and it's little d, little d. So these are all the different permutations of the genotypes between two parents that are both heterozygous for dimples. Now remember that having dimples is a dominant trait, so that both parents do have dimples. Okay, that's their phenotype. But the genotype is being heterozygous. They both carry a recessive gene. Okay, so now we can see the different permutations of the different children they can have. They can have a homozygous dominant child, big D, homozygous dominant meaning that they have both the same alleles and they're the same, big D, so it's homozygous dominant. They can have twice as many chance of having a child, big D, little d. This is heterozygous, just like the parents, big D, little d. Finally, they have a possibility of having a child that's little d. Now, since they're both the same, it's homozygous. Since they're little d, that means they're recessive. So we call this homozygous recessive. So they have a 50% chance of having a genotype that's heterozygous. They have 25% chance of having a homozygous dominant child and 25% chance of having a homozygous recessive child. So 50 plus 25 plus 25 equals 100%. So that's the genotype. The phenotype, we know that homozygous dominant, that big D is dominant, so big D, they're definitely going to have dimples. And then 50% of the children will be heterozygous, they will still have dimples. Only 25% of the children will have little d and will not have dimples. So this is the difference between genotype and phenotype. The genotype tells us the alleles present and which ones, and the phenotype tells us the physical expression. Okay. So we kind of went ahead of ourselves, but let's go back and go slower now if you didn't get it the first time. The Punnett Square shows possible genotypes of offspring from a man and woman, both heterozygous for dimples, big D, little d. Okay. The sperm and oocytes from each receive one allele and are haploid. Thus, each gamete may have either a big D, that represents dimples, the dominant trait, or little d, no dimples, or the recessive allele. Okay, so now I've circled it for you here. So here's the mother's genotype. Here's the genes she can give to her children. She can either give her a big D or a little d. Here I've circled the sperm from the father. It could either big D or little d. So there's an equal probability of giving one of these to their offspring. So when we did the cross in the Punnett Square, we've determined the probability of offspring with each possible genotype. So we said they were 25%, or 1 out of 4 children will be big D, 50%, or 1/2 the children will be big D, little d, and 25%, or 1 out of 4, will be little d. So these are the 3 possible genotypes they can produce, and these are the percentages or probability of having that genotype in the child. So here I circled them for you in the Punnett Square box. So big D is homozygous dominant. We have 2 big D, little d, that's heterozygous, and we have little d, homozygous excessive. So since we have 50% big D, 25% little d, and 25% big D. Okay, so that's how we determine the genotype and the probability of obtaining children with that genotype. How about the phenotypes? Now we can do the probabilities of the offspring with each possible phenotypes. Remember that the dimples is the dominant trait. Okay, so it's going to mask the recessive trait. So regardless if they're homozygous dominant or heterozygous, they will have dimples. So we can put these together. So 3 out of 4, or 75% of the children will have dimples. Okay, only 1 out of 4, or 25%, will have little d. That's homozygous recessive, and they will have no dimples. Okay, so 75% will have dimples, 25% will not have dimples. Okay, but for the 75%, we don't necessarily know the genotype. Okay, so here I've circled the phenotypes in the Punnett Square box. Okay, so we said that big D has dimples, big D, little d has dimples, but only little d, that's homozygous recessive, has no dimples. This again shows a complete dominant recessive control of the alleles. So whenever you have that dominant allele, it will express. Okay, and in order to get the recessive trait, you need both recessive alleles. So that was an example of complete dominance. So when again in complete dominance, whenever you have the dominant allele, it will express as the phenotype. You can only get the recessive trait in which you have 2 recessive alleles. Now let's look at a different example now where we have incomplete dominance. In other words, that both alleles can express, and we get what's called a hybrid. So the heterozygous genotype results in a hybrid phenotype. Okay. In plants, it's easy to see because you can have white flowers and red flowers, and you cross them and you get pink flowers. The same kind of idea happens in human genetics. Okay. So traits with incomplete dominance. An example of incomplete dominance is a gene causing sickle cell anemia. Okay, if you learned this in SCB 203 laboratory in the blood lab, in which one gene, and also in lab 12 of SCB 204, that one mutation of the beta globin gene turns the nice biconcave disc of a red blood cell to a sickle cell shape. And the result, it doesn't carry oxygen very well, it doesn't flow through blood vessels very well, and it's very painful. So the dominant trait here is normal. No sickle-shaped red blood cells, is big S. Little s is sickle-shaped. So we have 3 different types of genotypes we can have. We have big S, this is homozygous dominant. Right? No sickle-shaped red blood cells. We could have little s, homozygous recessive, which is sickle-shaped red blood cells. And we can have the heterozygous trait. Now remember, this is incomplete dominance, meaning that they both express now. So you're going to have both normal and sickle-shaped red blood cells. So our analogy with the flowers is not perfect, because in the flower you get the hybrid of the 2 colors. Here we don't get a hybrid of the shape of red blood cells, but we get both normal-shaped red blood cells as well as sickle-shaped red blood cells. So here's the genotype. Little s, big S, little s, and here's the phenotype. Okay. Again, we can do this Punnett Square, and we can show the mother and father. In this case, we're going to say they're both heterozygous for the sickle cell gene, big S, little s. In other words, they express both normal and sickle cell red blood cells. Okay, and we set up our Punnett Square just as we did before. Here we have the mother's genotype. She give the child one dominant trait, the big S, and she give one recessive trait to the child, the little s. The same thing with the father. In his sperm, he could donate one big S, the dominant trait, or little s, recessive trait. Okay. And if you do the cross, we're going to find that the probability of each genotype are 50% heterozygous, big S, little s, and 25% big S, homozygous dominant, or 25% homozygous recessive. So here's the cross now. So again, we cross, we get big S, we have big S, little s, we have big S, little s, and little s, little s. So that's how we get 50% heterozygous. In this case, we call this sickle cell trait. Okay. And then normal hemoglobin and little s is sickle cell anemia. So they have a 1 out of 4 chance to have a child that has sickle cell anemia. Okay, and so here's the probability. To have a sickle cell child, 1 out of 4, 25%. To have the sickle cell trait will be 2 out of 4, or 50%. And to have a perfectly normal child will be 1 out of 4, or 25%. So by using these type of genetic analysis, carriers of certain disorders can have a quantitative prediction of the probability of having a child with a disorder, which is kind of helpful. Okay. Now another type of inherited way of inheriting traits is called codominance. In this case, traits with multiple alleles involves traits with multiple alleles that are equally expressed, such as in the A, B, O blood group. Okay, so remember that in blood typing, you can either have blood type A, B, AB, or O. So that's called the ABO blood group. Okay, now in codominance, as the name suggests, that some alleles are equally dominant and equally expressed. Okay, so the dominant alleles for blood typing are big I capital A and big I capital B. These are the dominant alleles. Okay, so that you have these are going to mask the recessive alleles, and if they're expressed together, they will express together. As you might expect, big I capital A expresses for the A antigen, and IB expresses the B antigen. Now, little I is the recessive allele. Okay. So let's look at the different types of blood typing we can have with the different genotypes. So remember, we said that big I capital A is dominant. And so blood type A can have two genotypes. It can either be homozygous dominant, big I capital A, that's blood type A. Or you could have big I capital A, little I, which is the I little I is recessive. And so the big I capital A will mask the little I. So that will be blood type A. So both of these genotypes will express the phenotype of blood type A. Likewise, blood type B also has two genotypes. It can be capital I big B and capital I big B. That's going to be expressed B antigen. Or capital I big B little I. Just like up here with A, this will be expressed as the B antigen. So blood type A, blood type B, there are a total of 4 different genotypes. Now, what if you have capital I big A, capital I big B? Since they both equally express, you express both. And this is how you obtain blood type AB. Okay. Now, lastly, we have the recessive genotypes. When you have little I. When you have little I, then you have blood type O. Okay. And you have no antigens. So those are the genotypes of blood types, and they follow the codominance method of allele expression. So let's look at an example of codominance. We'll do the Punnett Square again, and we'll show a mother that's heterozygous for blood type A, meaning that she has one antigen, one allele that's big I capital A and little I. And the father that's heterozygous for blood type B. He has big I, capital B, and little I. Okay, so here we have the different alleles that they can express in the mother's ova, big I, capital A, and little I. And here's the alleles that can be expressed in the sperm of the father, big I, capital B, and little I. Okay, so these are the possible alleles they can give to their children. So when we do the cross with the Punnett Square, following codominance, we find there's 4 possible genotypes for offspring with equal probabilities. You could have big I, capital A, big I, capital B. You could have big I, capital A, little I, big I, capital B, little I, and little I, little I. And each one is 1 out of 4, or 25% probability of having one of these blood types. So, if we do the crosses again, this is how we get the big I, capital A, big I, capital B, the big I, capital B, little I, big I, capital A, little I, and little I, little I. So here are the 4 genotypes, and since there's 4 different ones in each box, each one has the equal probability of it being expressed, or 25%. So they can have an equal chance of having a child with these genotypes. Okay, now let's look at the phenotypes. Okay, that means for they can have a child of 4 different blood. They have the equal probability of having a child with all different blood types. Each one 1 out of 4, or 25%. So 20% have blood type AB, 25% blood type A, 25% blood type B, and 25% blood type O. Okay, and this is here, we've labeled them here. So a person that's parents that are heterozygous for blood type A and heterozygous for blood type B can have equal probability of having 4 different blood types. Okay. Another type of genetic inheritance is called sex-linked traits. Okay. Sex-linked traits are expressed on X or Y chromosomes. One example is Duchenne muscular dystrophy, colorblindness, or hemophilia are examples of disorders that are sex-linked traits. Okay. Now, sex-linked traits, the Y chromosome contains genes that determine the male phenotype. Males are XY and females are XX. Okay. Sperm carry the Y chromosome and determine the sex of the offspring. So if you have a Y, you're male. If you don't, then you're going to be female. Okay. Now, the X chromosome is a normal-sized chromosome, whereas the Y is a little dinky one. It's getting smaller and smaller and smaller. So the X chromosome has many genes not found on the Y chromosome. Okay. So here's a picture of the 2 chromosomes. Here's the X chromosome, and here's the tiny little Y chromosome. So all this part of the Y chromosome is missing from the Y chromosome. Now you can see that if you're a male and you have a defective gene on this X chromosome, you're not going to have the corresponding gene on the Y chromosome. So it's abnormal, then you're kind of in bad shape because you have nothing to compare it to, nothing to help it. If you're a female, you have 2 X chromosomes. If one is defective, you still have the other one that could be normal. So that's why that many sex-linked traits are expressed in males than females. Okay. So the X chromosome may have harmful alleles that produce X-linked disorders. Okay. Males often express traits for X-linked disorders because the Y chromosome do not have the corresponding allele to influence its expression. Okay. However, if you're a female, you can express a trait for an X-linked disorder only if she is homozygous for a recessive allele, meaning that she needs to have both alleles to be abnormal for her to express the trait. Okay. Females heterozygous for recessive alleles do not show the phenotype of the trait or carriers of that allele. In other words, it's more likely that a female will have one normal allele and maybe one abnormal allele. Okay, but it's in being masked, and so they are saying that person is a carrier of that allele. That she doesn't express it, but it's possible for her to give it to have it inherited from one of her offspring. Okay, and with sex-linked traits, we still use the Punnett Square. In this case, we're going to show a mother who is heterozygous for red-green color blindness, and here she has a normal gene, X capital R, but here's the color blindness, X little r. So she's heterozygous. So she's a carrier. And a father with normal red-green vision. So he has the normal X chromosome, X capital R, and his Y chromosome. And we do Punnett Square. So here's the mother's genotype. She can give an allele X big R and X little r. The father can give the normal X allele, X capital R, and his Y chromosome. Okay, and we do the cross, we'll get 4 possible genotypes of offspring with equal probabilities. We have big X capital R, big S capital R. This is homozygous dominant. That'd be 25% chance. Big S capital R, big X little r. This is heterozygous female. That's a carrier. That's 25%. You could have a male with normal vision. That's 25% chance. And a big X little r Y, a male that's colorblind. So they have a 25% chance of having these genotypes, each of these genotypes. And here they are in this Punnett Square. So again, we do the same thing. We take each allele and we cross them, So mother's genotype and the father's genotype, big X, big R. Here we have big X, big R, big X, little r. Here we have X, big R, Y, and X, little r, Y. Okay. So we've got 25% of each of these different genotypes. So what does this mean regarding the phenotype? Okay. Well, we have 25%. You could have a female that's 25% of normal vision, big X, X, big R, X, big R. You could have 25% chance with a female that's carrier but also has normal vision. That's X, big R, X, little r. You can have a male with normal vision, X, big R, Y. Or you could have a 25% chance of having a colorblind male, X, little r, Y. Okay, so here we matched up the phenotypes with each of the genotypes. Okay. So they could have, again, they could have 25% chance that's a carrier, 25% of a female that's a normal vision, 25% of a male that has normal vision, and a 25% chance of having a male that's colorblind. Okay. So when you think about it, though, if they're only interested in if they're colorblind or not colorblind, if they have a girl, the girl will have 100% chance of having normal vision. Okay. See, 100% of normal vision. However, if they have a girl, it has a 50% chance of being a carrier. If they have a boy, they have a 50% chance of a boy having normal vision and 50% chance of a boy having colorblind. Okay. However, if they're just thinking about their children in general, they will have 75% chance of having a child that's normal vision and only 25% of child colorblind. Okay, does that make sense? When you think about this, and it should come clear after a while. Okay, so in other words, they have no risk of having a colorblind female, but a 50% chance of having a red-green colorblind male child. Now, single gene traits are more rarer. In most cases, multi-gene control inheritance patterns, and we call these polygenic inheritance patterns. So most inherited traits are controlled by effects of 2 or more genes, such as eye color, height, and skin color. And to give you an example, we'll give you an example of a polygenic inheritance pattern. It can get complicated if it's a very large and many alleles. Okay. So let's look at height then. Height is regulated by 2 genes. The first gene has 3 genotypes. Big T is very tall. Big T, little t is moderately tall. And little t is short. Okay. The second gene that's controlling height has 3 genotypes as well. Big A reduces height significantly. Big A, little a reduces height slightly, and little a has no effect on height. And so if you do the Punnett Square, it's going to get a little more complicated now because we have 16 possible outcomes. So now our Punnett Square is a lot bigger. So now we have, since we have multi-gene traits, the alleles that can be given by the mother and the father there's going to be 4 of them now. Okay, so you can have big T, big A, big T, little a, little t, big A, little t, little a. Likewise, the father can give big T, big A, big T, little a, little t, big A, little t, little a. And now we do the cross of the Punnett Square as we did before, but now we have many more combinations. Okay, oops, and we can see now it does the same thing. I actually advanced my slide already, which I can't go backwards, but the same thing will affect. You have to look at the probability of the genotype and also the probability of the phenotype. Okay. Now, finally, we go to prenatal and newborn genetic screening, which can be done And the two types are amniocentesis and chorionic villi sampling. Amniocentesis, amniotic fluid is withdrawn using a needle inserted into the amniotic cavity between about 14 to 20 weeks of pregnancy. And some of the cells there, they can that come with it, they can analyze it for genetic defects. Chorionic villi sampling occurs during week 10 and 12 pregnancy, in which chorionic villi tissue is drawn through the vagina and uterus or by needle through the abdominal cavity. Again, this can be analyzed. Here's a really big needle here.